Hi folks, welcome back. And I guess it's welcome back for me too, because I haven't made one of these in like a month because I've had a cough. It just will not go away. But it's doing a little better. So let's see if I can get through this without coughing. If I do, I apologize ahead of time, but let's give it a go. Anyway, I want to move on to discussing the functions of DNA that are essential for life. And I want to start by reminding you of what those functions are. And to do that, we're going to use this slide. And here's the slide that we've used before. This is what I call the complete version of the molecular biology dogma, the one the evolutionists don't want you to see. And as a reminder, this is the version that they teach everybody in just about every science class. And like I've said before, this is intentionally and strategically left incomplete. Because once you learn the complete version, you realize that the dogma of molecular biology is impossible to explain scientifically. So now let me come back to the complete version here. And in this, we see that the process of transcription creates RNA from DNA, and the process of translation creates proteins from RNA. And the process of replication creates more DNA from existing DNA. And there are about 300 molecules absolutely required for these life-essential functions to occur. There's about 200 RNA molecules and about 100 proteins. Each one of them is required for these processes to go. And that's just to turn the machines on, so to speak, before you start making all the RNA and proteins required to create a species. So what I want to do now is go through some of these molecules and very briefly explain how these processes work. What I want to show you is that not only is the structure of DNA unique, its functions are too, since it governs and performs all the processes required for life. And I'm going to start here with transcription. Transcription is the biological process where DNA is used to make an RNA copy of itself. Transcription is the process of creating an RNA molecule that's complementary to a DNA sequence. RNA is a nucleic acid just like DNA, but RNA nucleotides utilize ribose instead of 2' deoxyribose. So let me show you this again. Here is a DNA nucleotide with the 2' deoxyribose sugar, which, like we talked about before, there is no O. There's no O at that 2' position right there. And here is an RNA nucleotide. And this one has the typical ribose sugar that has an O there at that 2' position. Therefore, RNA has ribose incorporated into its backbone and not 2' deoxyribose. RNA contains 3 of the 4 same nucleotides as DNA, with one exception, that being T. RNA does not use T. RNA utilizes uracil, which is U, instead of T, which has an almost identical structure with one noted exception. Here are the 4 DNA bases, which includes our T, and you see that CH3 right there. See that? If we go back to U with our RNA bases, the U doesn't have that CH3. So uracil U is missing the U, is missing that CH3 group that T has. So that's the difference between the bases used by RNA and the bases used by DNA. RNA is typically single-stranded as opposed to DNA, which is typically double-stranded. Now, there's exceptions to this rule, as in the case of some viruses that have a double-stranded RNA genome. But for the purposes of what we're discussing here, the dogma of molecular biology, we're going to focus on the single-stranded RNA. And transcription is primarily accomplished through the function of an enzyme called RNA polymerase II. DNA is not just an extensive series of A's, T's, G's, and C's placed haphazardly and repetitively throughout the molecule. There's order and logic to the placement of these 4 bases. There are numerous reasons and functions for the many arrangements, but for our purposes here, we'll focus on the arrangement of DNA into genes. A gene is a unit of heredity that contains functional regions of DNA, such as regulatory regions and transcribed regions. Genes contain the DNA blueprint that tells the cell machinery how to assemble a given protein via the messenger, the RNA. Although sometimes the RNA remains as a functional RNA and it's not made into protein. Through the process of transcription, the DNA template is copied into an RNA molecule that contains RNA bases in the identical order of the DNA bases. The one difference, however, is, as I said, RNA has U in place of a T. But to the process of molecular biology, they mean the same thing. The gene structure, or the DNA itself, also tells the RNA transcription machinery where to start and where to stop, as well as which genes to transcribe at which time. Eukaryotic genes essentially contain 4 basic elements: a promoter, an enhancer regions, untranslated regions, and translated regions. This slide shows an illustration of the structure of a eukaryotic gene. Promoters are a DNA sequence that functions as a signal to RNA polymerase II that the beginning of a gene sequence is very close. RNA polymerase II binds to the promoter region DNA to initiate the process of transcription. Promoters contain a region called a TATA box, the region where RNA polymerase II starts to open up the DNA to gain access to the bases it will use as the template to transcribe the RNA molecule. Enhancer regions are DNA sequences that bind proteins, transcription factors, that are used to enhance the level of gene transcription. Untranslated regions are those that are transcribed into RNA but not translated into proteins. Untranslated regions within the transcribed region are called introns because they intervene, interrupt, and divide up the transcribed region. The introns vary in length within genes and from gene to gene. The untranslated region, found upstream of the transcribed region, is called the 5' untranslated region, or 5' UTR. While the untranslated region found downstream of the transcribed region is called the 3' untranslated region, or 3' UTR. These regions function in post-transcriptional modification and regulatory process that act on the RNA. Finally, the transcribed region contains the actual DNA sequence that will be transcribed into an RNA molecule. The transcribed region contains both translated and untranslated regions. The translated regions are called coding regions because they code for a protein sequence and are translated into that protein. They're divided up into sequences called exons, which vary in length just like the introns. The exon boundaries are defined by the presence of the introns and vice versa. The exons and introns create almost a checkerboard pattern when depicted in an illustration. So the transcribed region contains the coding regions, the exons, as well as the non-coding regions, the introns, the 5' UTR, and the 3' UTR. So when we're looking at this slide, as you've been following along, I have all of those regions highlighted in this illustration. Estimates of the average size of a human gene structure ranges from 10 to 15,000 base pairs up to 30,000 base pairs. Now, this is only an estimate of the average size gene, and gene size varies greatly. The average size of the final messenger RNA, the mature mRNA that's used for the process of translation into a protein, is only 12 to 1,500 bases. The human genome contains about 23 to 25,000 unique protein coding genes. In fact, only about 1.5% of the human genome codes for proteins, while the rest consists of RNA-only genes, regulatory sequences, repetitive elements, and non-coding DNA. Now, if you look at this slide, we see an illustration of the process of transcription. RNA polymerase II reads, that would be that guy right there, this guy right here. RNA polymerase II reads along the open DNA on what's called the antisense strand, which is this bottom strand down here, and creates an RNA molecule that's identical to the other strand, the coding strand. So you see, as it's doing this, it's using this strand as the template, but the strand that it's making is going to be identical to that DNA strand up there. But it can't do it alone. It needs the help of other proteins, which are shown here, which are collectively called the transcriptosome, which I'm showing you in this bunch of proteins over here. The major proteins that comprise, they don't compromise, shall we say comprise, that's a better word, a transcriptosome include RNA polymerase II, the one that actually makes the RNA, TATA binding protein, which is a subunit of transcription factor 2D, which is the first thing to bind the TATA box of the DNA. Then we have 5 more proteins called transcription factors that bind the promoter, and they then mediate the binding of RNA polymerase II to these proteins. So these other proteins lay down and bind on the DNA and kind of make a little nest for RNA polymerase then to bind there. Transcription factor 2H, one of these 5 transcription factors, this one has helicase activity. And what that means is it can separate the 2 DNA strands by breaking the hydrogen bonds between the bases. So as I'm showing, the RNA polymerase has got to get in here. So the transcription factor 2H separates those 2 strands of DNA so the RNA polymerase can get in there and read, as in this case, the strand on the bottom. So when it does that, you see it kind of looks like it's a little bubble. And that's exactly how it looks. It forms a little bubble in the DNA so the RNA can read the bases. Then there's also activators and repressors that are responsible for driving the rate of transcription. Because on its own, RNA polymerase II only has a low rate of transcription. It needs these other factors to boost that rate higher. And there are also RNA editing factors that proofread the growing RNA strand to ensure its accuracy. Now, eventually, RNA polymerase II will reach a stop sign in the DNA structure of the gene, and it will stop transcribing. The RNA will fall off the DNA, and the DNA gets closed back up again. The RNA after that then receives some modifications. The 5' end is capped. They call it the 5' cap, which is a modified G nucleotide. And there's a cap synthesizing complex that makes that happen. The 5' cap protects the RNA from degradation outside the cell nucleus. The 5' cap is also a signal for the ribosomes to attach to it and initiate the process of translation, which we'll talk about next. Now, the 3' end received what's called a poly A tail, where anywhere from 30 to 250 A nucleotides get added to the end of the RNA. There is an enzyme that makes that happen called polyadenylate polymerase, and they call that process of doing that polyadenylation. The poly A tail helps prevent degradation of the RNA. It assists with the passage of the RNA from the nucleus into the cytoplasm of the cell, and it acts like a translation termination signal. Now, the interior portions of the RNA, which at this point is called pre-mRNA, they undergo modifications as well. And this slide is depicting how this works. The process of RNA splicing is used to cut out the non-coding regions of the pre-mRNA. As the non-coding bases of RNA, representing the introns, are cut out, the coding bases of the RNA, represented by the exons, are brought next to one another in tandem and then spliced back together. The introns have special base sequences at both ends that signal where they're to be cut out. And the splicing of the pre-mRNA is accomplished with the use of a spliceosome. Spliceosomes consist of units of small nuclear ribonucleoproteins, which are composed of RNA molecules and proteins. They are these small nuclear ribonucleoproteins are RNA molecules that act as catalysts for the cutting and splicing process. So I'm trying to show you those guys up here, right? And they're kind of like, for lack of a better term, a big pair of scissors. So they will cut out the introns there and there, right? And then all those exons get brought together and put in tandem, all in a row with the introns missing. So after we have the 5' cap added, the 3' poly A tail, and we've cut out the introns, the pre-messenger RNA is now considered a mature messenger RNA, what they call mRNA, ready to be translated into a protein. These modifications of the mature mRNA are recognized by transport proteins, which escort the mRNA out of the nucleus and into the cytoplasm through holes in the nucleus called nuclear pores. And so, folks, that in what I've just shared with you, I've given you about a semester's worth of molecular biology in just about 10 minutes or so. And I left a ton of stuff out, as you can probably imagine. But anyway, here's the point of me doing this. These molecules that I just discussed that are part of this process, they're all part of those 300 or so needed for the life essential processes of molecular biology. You should be able to see that all these molecules are critically important for all these processes, and therefore they are critically important for life. Without any of these molecules, with any of them missing, the critical parts of these processes that they're responsible for, they won't happen. And if any of these steps in the processes don't happen, life can't happen. Therefore, as I've already explained, all of these molecules have to be here at the same time, functioning together, or life cannot exist. They cannot accumulate gradually over time because they're all needed from day one. And what this is, folks, is what you call irreducible complexity to the utmost. And this is a topic that I'm going to address further. But for now, I need to end this video, and we'll save that for the next videos. So come on back for that. And if you like what you hear, give me a thumbs up and subscribe to my channel. Thank you very much for watching. And as always, we'll leave the light on for you.